Hi everyone. In today's video, I wanted to go through the neuroglial cells that are present in the central nervous system. If you haven't already seen it, make sure you watch my video on the neuroglia in the peripheral nervous system, as many of the neuroglial cells in that system relate in their function to this system. So, the central nervous system. It contains 4 different types of neuroglial cells. Firstly, there are ependymal cells, astrocytes, oligodendrocytes, and microglia. So in this video, I'm just going to go through all 4 of them and talk to you a little bit about their function. So let's start off with number 1, ependymal cells. Ependymal cells are actually cells that are present in the ventricles of the brain, usually on the roof of the ventricle, and they are responsible for the synthesis, and these are keywords, the synthesis, filtration, and movement of cerebrospinal fluid within the ventricles. So all of these functions relate to the CSF. Now these cells are actually present on what we call the choroid plexus. The choroid plexus is basically a structure of capillaries that contains these ependymal cells on the outside, and as blood plasma is able to flow through the choroid plexus, it is able to be essentially filtered. So as the ependymal cells contain special ion channels on their surface, they're able to allow for the filtration of the blood plasma so that it can be essentially converted into cerebrospinal fluid to then be circulated around the ventricles in the brain. So these cells are somewhat present as a lining, and they contain what we call stereocilia. And these stereocilia can be likened to elongated microvilli. However, unlike microvilli, in which the function of microvilli is to allow for the absorption of nutrients, for example, in the small intestine, stereocilia in this case are able to allow for the movement of cerebrospinal fluid by various motions around the ventricles in the brain. So essentially, their own movement is able to allow for a function similar to a rudder on a ship, and therefore allow for the movement of the cerebrospinal fluid in various directions. The other glial cell that I wanted to talk about today is the astrocyte. So astrocytes, if you had seen my video on the peripheral nervous system, are analogous in their structures to satellite cells in the peripheral nervous system. However, there are some key differences in their structure that exist. So, for example, say that this is the lumen of a blood vessel or a capillary within the brain, and this being the endothelial lining. Now, astrocytes, unlike satellite cells, which directly are in contact with the soma of the neuron, actually have perivascular feet or false feet that attach to the wall of the capillary and extend towards the neuron. So we have our neuron here with its body, dendrites, and axon terminal. So these astrocytes are somewhat like spiders in that they allow for their small feet to extend onto the appropriate capillary. And this role has 2 main functions. Firstly, because the neuron is separated by this barrier, it is essentially able to allow for the diffusion of nutrients across this barrier to the neuron. However, most importantly, the astrocytes are actually able to allow for the stimulation of tight junctions between the endothelial cells on the capillaries. Now, capillaries are actually vessels that contain one or two epithelial cells. So imagine this being one cell and this being another. In this case, an astrocyte would allow for the formation of tight junctions between the cells. This ensures the formation of a very important structure called the blood-brain barrier. Abbreviated to BBB. Now, this blood-brain barrier is a very important function in the brain because it essentially allows and acts as a filter in the brain to allow for the filtration of unwanted substances, and these could be various pharmaceutical drugs or other molecules which may be possibly harmful for the brain. So the blood-brain barrier is formed, or the tight junctions that form the blood-brain barrier are stimulated for their formation by the perivascular feet of the astrocytes and therefore allow for the protection of the brain. This also assists in the function of the astrocytes to allow for the diffusion of nutrients, as neurons are quite delicate and in some parts of the nervous system are actually unable to be remade if they are or repaired if they are damaged. So therefore, this astrocyte plays a very important role in the central nervous system. The third glial cell that I want to go through today is the oligodendrocyte. And if you watched my previous video on the peripheral nervous system, you'll notice that this structure is actually analogous in its function to Schwann cells in the peripheral nervous system. So similar to Schwann cells in the peripheral nervous system, these oligodendrocytes, and similar to astrocytes, have their own cellular projections that project from the cell body of this particular glial cell and attach to the neurons in the central nervous system. So, if I draw an example of a particular oligodendrocyte here, it will have up to 50 to 60 projections that extend from its body. And these projections essentially allow for the process of myelination to occur, similar to Schwann cells in the peripheral nervous system. And this myelination, as I mentioned in my previous video, allows for, firstly, the protection of the neuronal cell body, as these myelination processes are 90% lipid and 10% protein. And secondly, it allows for the process of saltatory conduction, which is when, through the formation of nodes of Ranvier, the nerve impulse is able to essentially jump across the axon in order to allow it to flow in a faster manner. So this is the function of oligodendrocytes. The last neuroglial cell that I wanted to go through in the central nervous system was the microglial cell. This microglial cell is similar in its function to macrophages in the rest of the body. However, their smaller size makes them quite important for the tight spaces and various structures that are present in the brain. Their function is almost identical to macrophages in that they are able to essentially ingest any cell debris, any wastes, or if there are any pathogens present in the brain, and essentially work as the janitors of the brain. These microglial cells are containing many different processes, like so. And these processes allow them to assist in their far-reaching capabilities in collecting various wastes. So these cells essentially are able to ingest and kill anything that may be harmful for the brain. So in summary, the 4 neuroglial cells present in the nervous system, specifically in the central nervous system, are ependymal cells, astrocytes, oligodendrocytes, and microglia. Ependymal cells are present on the choroid plexuses and are responsible for three main functions: the synthesis, filtration, and movement of cerebrospinal fluid within the ventricles of the brain. They have this purpose by allowing for ions to pass through from the capillaries of the choroid plexus and have these filtered by ion channels, and movement is able to be allowed for by the stereocilia, which are cellular projections that are able to allow for the movement of the fluid throughout the ventricles. Secondly, there are astrocytes present in the central nervous system. These astrocytes are also a very important neuroglial cell, as they are essentially able to allow for the formation of the blood-brain barrier, and also they allow for the diffusion of nutrients from the blood vessels that pass into the brain into the neurons, therefore stopping any pathogenic or possibly harmful substances from affecting the brain. Thirdly, we have oligodendrocytes, which are analogous in their function to Schwann cells in the peripheral nervous system, and their processes allow for the process of myelination to occur. Myelination therefore allows for the protection of the neuron and also saltatory conduction. And finally, we have microglial cells. These are essentially the janitors of the central nervous system in that they allow for cleaning up of any cell debris or wastes within the brain, and also allow for the phagocytization of various pathogens that may be present in the brain or spinal cord. So, this has been all of the different neuroglial cells in the central nervous system. I hope you've enjoyed the video. Thanks for watching.